Delivering anything therapeutic to the brain has long been a challenge, largely due to the blood-brain barrier, a layer of cells that separates the vessels that supply the brain with blood from the brain itself. Now, in a study published August 12 in Nature Biotechnology, researchers have found that double-stranded RNA-DNA duplexes with attached cholesterol can enter the brains of both mice and rats and change the levels of targeted proteins. The results suggest a possible route to developing drugs that could target the genes implicated in disorders such as muscular dystrophy and amyotrophic lateral sclerosis, ALS. It's really exciting to have a study that's focused on delivery to the central nervous system, with antisense oligonucleotides given systemically, says Michelle Hastings, who investigates genetic disease at the Rosalind Franklin University of Medicine and Science in Chicago and was not involved in. The authors showed that it works for multiple targets, some clinically relevant. See Getting Drugs Past the Blood-Brain Barrier in 2015. Takanori Yokota of Tokyo Medical and Dental University and colleagues published a study showing that a so-called heteroduplex oligonucleotide, HDO, consisting. In the new study, the research team showed that an HDO designed to target a tumor-associated long non-coding RNA, MALAT1, achieved a higher level of knockdown in the brain and spinal cord when tagged with cholesterol and injected intravenously than when the HDO was tagged with the lipid tocopherolin. In both rats and mice, the effects of the knockdown were tied to the dose of the HDO, and 4 doses, given a week apart, were most effective. An intravenously injected single-stranded oligonucleotide tied to cholesterol didn't knock down MALAT1. The data seems to suggest that the single-stranded oligos may get trapped and may not actually cross the vasculature as efficiently as the double-strand, he explains. The targets of the other 2 HDOs demonstrated more modest knockdowns. The data seems strong. Rudy Giuliano, an emeritus professor at the University of North Carolina School of Medicine who did not participate in the study, writes in an email to the scientist. Open questions, he adds, include whether most of the effect observed in the whole brain actually took place in neurons and not in supporting tissues, if the high doses the authors used, 50 mg of HDO per kilogram of body weight, can be toxic, and why this simple modification of oligostructure. You might want to knock down inflammation in the brain in, say, multiple sclerosis. You're probably going to have to be giving quite sizable doses on quite a long-term basis if you're just giving an oligonucleotide, which doesn't effectively regenerate itself. In the study, the authors acknowledge the limitations of the current work, and Rigo says plans for optimization are already. Theoretically, you would want to identify how those lipids are getting into the brain. What receptors and what systems are they using? He asks. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.